All right, everyone. Welcome to the HudsonAlpha BioTrain Internship presentations. We're really glad you were able to join us today. My name is Michelle Morris, and I lead our BioTrain Internship program. This is our 12th summer to do BioTrain internships, and we plan to have 27 students here on campus participating in positions in our labs, our administrative departments, our associate companies. But when it became clear that we weren't going to be able to host these on-campus experiences, I asked our mentors who might be able to transition and lead their intern virtually in a virtual remote position. And we were very fortunate to be able to have 10 positions that we could transition to this remote type of research, primarily because these positions were computational in nature. So I'd like to say a really big thank you to all the mentors that were able to take on that challenge. These interns working remotely have done some really amazing research this summer, and we're really happy to be able to share it with you today. So before we get started, a couple of things. First, please note that the webinar is being recorded for those that have registered and weren't able to attend, and soon we'll be able to post this video up on a HudsonAlpha website for everyone to see later if they'd like to. Next, I'd just like to say that each intern will present and then have just a couple of minutes to take your questions. So we welcome you to type your questions into the Q&A box. If you look down at the bottom of your screen and maybe you need to move your cursor over the bottom of your screen, a toolbar is available and there should be an icon for Q&A. So if you have a question for one of the interns, just open that icon and type in your question, and after they finish presenting, I will read your question out loud and give them the chance to respond. We are trying to keep the presentations kind of on track and not go too long today, so they will only have just a couple of minutes to take questions. All right. First, I'd like to start with Ashlyn Anderson. Ashlyn is from the University of Florida. She worked with Dr. Greg Barsh and on an evolutionary bioinformatics project, and also with Brenda Larison at UCLA as a collaborator. So, Ashlyn, go ahead.
Hi, I'm Ashlyn. I was an intern in Dr. Barsh's lab this summer, and I was looking at the underlying genetic basis for striping in zebras. So, next slide, please. Sorry. So there is still a lot that is not understood about the genetic causes for striping in zebras, but by studying their patterning, we can try to get insight into the genetic architecture for the cause of striping. To do this, we can use zebras that naturally show variation in their striping patterns in order to assess the, same slide, the genetic differences between the groups. A population of zebras in Rwanda is therefore a good candidate for identifying genes related with striping because the population contains individuals that are striped and rare individuals that are spotted. There's a range in phenotype in the population. There can be a slight amount of spotting up to more extensive spotting and fading like in the individual on the top right. Next slide. So to look at the difference between the two groups of zebras, the spotted and the striped zebras, I used two types of measurement. First, I measured heterozygosity. And because all of the data came from low coverage sequencing, sequenced at a coverage of 2x, heterozygosity was assessed by a pooled allele count and not for each individual in the population. I also used the fixation index, or FST. And as you can see from the plots, the distribution of heterozygosity was similar across the two groups, indicating that the two groups have a similar level of diversity. And since the striped and spotted groups are from the same population, it allows me to further investigate the genetic differences between the two groups. So for the selection scans, I was looking for regions of low heterozygosity because it might indicate regions necessary to expressing patterning. And I was also looking at regions of high FST because it tells us which regions are fixed in the spotted population compared to the striped. And the goal was to find regions of the genome that overlapped with low heterozygosity and high FST. And we could be looking at multiple significant regions because we don't know yet if striping is a monogenic or polygenic trait. Next slide. Yeah. So then I performed these selection scans comparing the striped and the spotted groups. The plot on the top shows FST, where the peaks are windows across the genome that have a significantly higher FST. And the plot on the bottom shows the difference in heterozygosity, so regions that had significantly lower heterozygosity in the spotted population than in the striped. As you can see, there were 3 large peaks that correlated between FST and heterozygosity that occurred on chromosome 1, 3, and 14. Next slide. So next, I wanted to assess whether the peaks I was seeing in the data were real significant regions, and I also wanted to compare them to a scan where there should be no difference between the 2 groups. So I permuted the data and randomly assigned an individual to either striped or spotted, then performed the selection scans. I did this for 100 permutations, then compared the true data set to the permuted. The permutations allowed me to assess the validity of the results in the true data, because if the permuted data appeared similar to the true data, then there was likely a high false positive rate. Next slide. So when looking at each individual permutation, there are still peaks that occurred across the genome, and these peaks are randomly distributed for each of the permutations. So this figure shows box plots for the number of significant windows in each of the permutations for FST, heterozygosity, and the overlap between the 2. So significant windows are defined as those that had a p-value less than 1 × 10⁻⁻⁻⁻, and the red lines are the number of significant windows that I found in the true data set. So you can see that for FST, there were a few permutations that had a higher number of significant regions than the true data. And for heterozygosity, you can see that the majority of the permutations had a higher number of significant windows than the true data. So this shows us that there's likely a high false positive rate for each of the measurements. So what distinguishes the true data from the permuted if the permutations see just as many peaks as the true data? In the true data, you see a lot more overlap in significant FST and significant heterozygosity windows. So the majority of the permutations had absolutely no overlap between significant FST and significant heterozygosity. I also looked at the relationship between FST and heterozygosity and got an R² for each of the permutations, as well as the true data set. And the mean R² for the permuted was significantly less than the true R², with no individual permutation having an R² that came within 0.1 of the true value. Next slide. So in conclusion, I still saw peaks across the permutations that are likely false positives. And since the correlation between FST and heterozygosity was what distinguishes the true data from the permuted, the regions of interest are those that overlap between the 2. So next, I need to further investigate the genes in these regions. I'm also starting to evaluate the effect of variants may have in these regions. And I also want to look at another population of spotted zebras to see if they share potential variants. Next slide. And I just want to thank everyone for an awesome summer, and thanks to everyone that assisted.
Great, thank you so much, Ashlyn. So this is your opportunity, if you have a question for Ashlyn, to go ahead and type that in our Q&A box. And as I mentioned, I'll be happy to read that out loud to our presenter and give them the chance to reply. And you can, of course, ask a question later. We can certainly go back to an intern if you think of something later. Okay. So if we don't have any questions, I'm going to move on to our next presenter. I'd like to welcome Bernie Mousa. Bernie's from Auburn University. He worked with our clinical services lab under the doctors Elaine Lyon and Matt Holt.
Hi everyone, so I'm Bernie Mousa, and I was a BioTrain Intern in the clinical services lab. And my project over the summer had to do with pre-alignment coverage using FAST-Q metrics. Let's go on to the next slide. So the overlying problem that I was looking at was how to reduce the time determines if coverage of an alignment is bad or not. So let's just look at the process overall. So you begin by collecting data and samples and placing them to a sequencer. The sequencer takes about 2 days and outputs a FAST-Q file. And you check this FAST-Q file and verify that its metrics are of a certain quality. It takes about 30 minutes. Next, you go to alignment, and that takes about 6 hours, produces a BAM, and essentially you're looking to see that the coverage was greater than or equal to 30x. After that, you go to variant calling, takes about an hour, output to VCF, verify its quality. So go on to the next slide. So my real focus was on alignment and that 6 hours and how to reduce that. Because if coverage is less than 30x, you have to do a top off, which just means I have to get more samples, more data to go back on the sequencer, to go through this process again, to go through alignment again, to hopefully get the coverage that I need. So the question is, can I short circuit this process? Can I go from FASTQ to sequencing? Let's go on to the next slide. So can the metrics from a FASTQ be used to determine the coverage before performing alignment? Next slide. So to do this, I used machine learning, specifically the scikit-learn library for Python. So by taking these various models that exist, you can train them using samples that exist in the clinical lab, split these samples up into a training set and a testing set, retrieve the FASTQ metrics that you can see on the right, and retrieve the coverage values and train these models to accurately predict coverage values. So go on to the next slide. So these are the results. N equals 137, that's 20% of the total data I had available to me. So you train using the 80%, which creates the models, and these are the results I received by using my testing data. So the graph in the top left is representative of predicted versus actual values. So that red line is where predicted equals actual, and the blue dots are representative of each sample. So if the blue dot is above this red line, that means it was an overestimate, and below it was an underestimate. The graph below that shows the actual difference between predicted and actual, and again, if it's above 0, it's an overestimate, and below 0 is underestimate. The graph to the right is called a Violin Plot, and it shows essentially the variations between the predictions. So I just took the deltas and plotted them, and it allows me to compare different models to see which one performs better. So for example, you can see with that first model, Random Forest, it had a prediction of 7 over what the actual value should have been, which is not what you want to see, not very accurate. If you look at gradient boosting in the middle, the highest value that it was off by was approximately 3. So that's a relatively good estimate, a good prediction, and gradient boosting ended up being one of the best models that I worked with and the one that really stuck with when I actually performed predictions. So I'm going to the next slide. So in conclusion, alignment coverage can be predicted fairly accurately. The best feature I saw was total bases, and that's just representative of how many bases I was able to retrieve from sequencing that could be used for further alignment and such. And my prediction script allowed me to potentially save 6 hours per sample, because it takes 6 hours to run through the alignment process to see if its coverage was up to par or not. So 6 hours saved by running a 20-second script is pretty solid. And there were a few cases of over and underestimate, but this can be accounted for by adding and subtracting standard deviations. So you can see on the right, I could subtract a standard deviation from the predicted coverage. And if you set like a threshold, say my threshold was 29, and that lowest standard deviation, 28.4, is below 29, then I would immediately perform a top off and just go through sequencing again, as opposed to going through the alignment process. Let's go on to the next slide. So going forward, my mentor, Matt Holt, you can see on the right, will be taking this and incorporating it into the clinical workflow, helping to make it easier to use, and hopefully reporting samples that require top off to make the process go a lot faster. Also, it could be expanded by additional modeling. You can train using more samples, because the more samples you use, the more accurate your models tend to be. You can also test with other machine learning models and see if there are better ones out there that possibly perform better than ones I tested, and maybe including other metrics, other features that are out there that could improve the accuracy as well. Next slide. So I would like to acknowledge these people and the work they put into making the summer happen, all things considered, and it's been incredible, and I've learned quite a lot. So that's all. Are there any questions?
Thank you, Bernie. We don't have any questions right now. Again, I invite you all, if you have a question, to type it into that Q&A box. So we're going to go on to our next presenter as we're waiting. In case anybody might have a question for Bernie, we can certainly hop back to him. I'd like to introduce you to Chelsea Owens from Jacksonville State University, who worked with our genetic counseling team. Chelsea was mentored by Kelly East and Megan Cochran.
Hi everybody, my name is Chelsea. Can everyone see me okay? Okay. My project was to measure the clinical utility of whole genome sequencing at the Smith Family Clinic for Genomic Medicine. Just to briefly touch on what clinical utility is, it's the relevance of a specific clinical test to a patient and how their doctor or specialist will utilize those results from that test. And insurance companies typically will not cover or reimburse the patient for that test unless there is evidence of clinical utility. So if you look at an x-ray, for example, x-rays have a specific health outcome. So those are more likely to be covered by insurance companies, whereas whole genome sequencing, it doesn't have as much clinical utility as the x-ray because it's not necessarily about treatment. It's about what further steps can we take for the care of the patient. Next slide. I had the opportunity to utilize a tool developed by researchers at the Hospital for Sick Children in Toronto, again, to measure evidence of clinical utility in whole genome sequencing. The tool essentially was a survey where I went through past patient files at the Smith Family Clinic and essentially translated them into the tool. And all of our empirical data you will see later is directly reflecting the patient files that I analyzed. The tool monitors these three things, and you can go to the next slide. So primary findings are genetic variations found that could be linked to the patient's health issues. Sometimes results do come back negative, and I know clinical utility is all about usefulness, but that doesn't make the negative result completely useless, as genetic research is still underway for a lot of diseases. This patient, if they do have a negative result, could mean that they have a genetic condition that has not been identified yet, and that's perfectly fine. In that scenario, we typically request that they come back in 18 to 24 months to get retested. The tool measures whether the primary finding provided an explanation for the patient's symptoms or, next, any of the following subsections. The more questions that were answered, the more data was provided that can increase the evidence of clinical utility. Next. And this graph shows that half of our patients provided no genetic explanation. Like I said earlier, those are the conditions that probably have not been discovered yet. And the other half provided complete, partial, or possible genetic explanations. Complete genetic explanations kind of speak for themselves. Partial genetic explanations are variants that were found that have some overlap to the patient's phenotype, but not all. And the possible genetic explanation are variants that are of uncertain significance, meaning that they have a very limited amount of information that could be just so happen to overlap with the patient's phenotype. And in order to increase the diagnosis from possible genetic explanation to a complete genetic explanation, either further testing or more clinical testing has to be done to confirm the diagnosis of the variant of uncertain significance. Next. Secondary findings are genetic variations unrelated to the current phenotype but could potentially be useful in the future. Secondary findings indicate one of two things. One is carrier status, which we will talk about soon, and the other one could be a variant found such as the BRCA1 mutation, which is linked to ovarian and breast cancer. So the patient may not be affected at the time, but could eventually develop the disease. Most everyone receives at least one secondary finding, unless of course notified by the patient. They can choose to not know any secondary findings. Most of these findings indicate carrier status for the patient and the patient's family. Carrier status simply means that the patient is a carrier for a specific disease. And looking at all of the files from past patients, a very common variant that I seem to come across really often is the Hfe variant for hemochromatosis. So say my patient has that variant, and they wish to have a child with someone else that has the Hfe variant for hemochromatosis, they would have to consider that the fact that they both have that variant means that their child is at a 25% increased risk to develop hemochromatosis later in life. And when untreated, hemochromatosis could be deadly. So it's good to know. Next. You can click one more. Both of these charts reflect that the patient files reflected information about recurrence risk for the patient and the patient's family, regardless of whether it's relevant now or in the future. And as you look at the recurrence risk information for the patient's family, 40.5% of the files reflected that the recurrence risk information cannot be determined. This is just because unless the patient opted to have duo or trio whole genome sequencing, the recurrence risk information cannot properly be reflected in the patient's family. Next. And pharmacogenomic findings, finally, are the variants that may affect the patient's metabolism of certain medications. A variant that I have seen alters the metabolism of warfarin. This does not mean that the patient does not need to take warfarin. It just means that they have to consider a different dosage. And the dosing guidelines are available to guide dosage of the drugs in the future. Of course, at the discretion of the doctor or the specialist, not at the patient's discretion. Next. 100% of the files did provide information to guide medication management in the future. Next. The data that I collected will be utilized in a manuscript that will be published this fall. I'm really excited about this. I get to work in collaboration with some really great people. Our main objective is to just increase and quantify all of the reasons why whole genome sequencing is important for the benefit of a very large group of people, not just individuals. And I have definitely experienced firsthand how this tool is very practical and neat, and I can't wait to see how it will influence genetic medicine in the future. Next. A special thanks to the following people for making my summer really, really enjoyable. I've had a great time.
Thank you, Chelsea. I do have a couple of questions. So Greg asked, do you have any data correlating the cost of the sequencing versus benefit to the patient or cost of care over the complete population of patients?
I do not have any data for that. That is a really great point, and I will definitely look into that. Thank you for pointing that out.
Thank you, Greg. And our next question is from James. Hey, Chelsea, I'm not sure how many patients you reviewed, but did you get to analyze these statistics with respect to time? For instance, did you find more things reported in patients analyzed more recently?
Yes, actually. In past files, there were several variants of uncertain significance that I did come to notice were actually diagnostic in patients that were tested, you know, say a couple months ago. So yeah, if I didn't answer your question, please let me know.
Thank you, Chelsea.
Thank you.
All right, we will move on ahead to our next presenter. I apologize, I didn't delete Ashlyn's slide, but now I have it correct. Ivy Gentry worked with our educator professional learning group. Ivy's from the University of Alabama in Birmingham, and we're happy to welcome her today to tell you about her experiences this summer.
Hi everyone, I'm Ivy. I worked for the educational outreach team here at HudsonAlpha. One of the responsibilities of the education team at HudsonAlpha is creating materials that make it easy for K-12 educators to teach genetic science topics in their classrooms. These include teacher backgrounds, student backgrounds, directions, materials, and worksheets. So I worked on writing and editing, teaching manuals covering lots of different topics, including DNA phenotyping, genome editing, and polygenic risk scores. So today I'm going to show you the evolution of a piece of the polygenic risk score manual, which I spent approximately the first half of my internship working on. Next slide, please. This is basically how teaching manuals begin. This was one of the ideas I was given that sparked a lot of edits, but it just starts as like a rough outline, and we go from there and we go through rounds and rounds of edits. So let me show you where I started. Next slide, please. So this was my first draft. I'll give you a second to read it over. If you have any background or experience in genetic or genomic sciences, you can probably understand the gist of what I was trying to say. Unfortunately, the sentence includes a lot of jargon, a few mistakes, and far too many words. The first round of feedback I received from my mentor reflected all of these notes. Next slide. So this was after my first edit. First, I wanted to incorporate the definition of monogenic to set up a contrast for the definition of polygenic. Next, if you noticed in the first draft, I misused the word genes. This type of mistake can lead to a lot of confusion and is the exact reason why the first draft is never what's published. Finally, I added a clarification sentence at the end to make it very apparent what polygenic referred to. I submitted this for another round of feedback, feeling really confident, but no worries, we made lots of changes again. Next slide. One thing we try to do with teaching manuals is align the reading level to the grade level the lesson is intended for. This isn't necessarily to make it easier for the teacher. We want the teacher to read the manual and immediately think, yes, my students can accomplish this. If we write manuals at college graduate level and make it more difficult to understand, the teacher will be less likely to consider the lesson plausible for their students. Some ways we do this is by reducing the number of 3 or more syllable words. So you'll see that I replaced the word however with though. And that's a simple example, but we try to do that throughout the entire manual. Another way is reducing the average number of words per sentence. This makes information more bite-sized and makes an easier flow. We also work to weave ideas through the writing so that when we get to the main point, the concept has already been planted. I also tried to reduce jargon by replacing continuous distribution of phenotypes with wide range. We were able to eliminate 2 multisyllable words and create a more user-friendly terminology. Finally, I tried to eliminate as many personal pronouns as possible to increase professionalism. All these changes led to the version that we have today, though surely it won't stay this way. This still requires edits from multiple teams, multiple groups throughout the team. So let me show you where we're headed. Next slide, please. These 4 sentences that we've reviewed over the past couple of minutes are just a small portion of one section of the teacher background. The teacher manual, as it stands now, is 8 sections and 7 pages long. So you can imagine I spent a lot of time writing and rewriting this manual. This teaching manual will also be paired with other materials in the module, including student background, worksheets, student instructions, and meticulously designed materials. So you can see that the education team does a lot of work to prepare our teachers for teaching their students what we are learning about currently. So I just wanted to thank the following people. Oh, sorry, next slide. I just wanted to thank the following people for such a fantastic summer. If you will go to the acknowledgement slide, please, ma'am.
I don't have an acknowledgement.
Oh, sorry.
Sorry.
Sorry, no worries, no worries. I wanted to thank the Educational Professional Learning team and Hudson Alpha for such a fantastic summer. And if you have any questions, please feel free to reach out.
Thank you so much, Ivy.
Of course.
And of course, we would welcome all your questions. We always enjoy our interns in educational outreach. Wish we had been able to see you face to face this summer. So. All right, I do have a couple of questions, Ivy, that we've been posted. Elaine asked, did you define traits before this paragraph?
So in this manual, I don't believe we did. This manual is intended for 9th through 11th grade students. So traits is typically considered age-appropriate terminology at that point. So no, I don't think we did, but it is kind of implied throughout the manual.
Okay. And James asked, Ivy, this was really informative for how manuals like this get built. Are there any tools available to highlight some of these things, like the word links, or is all of this manual?
Yes, so there's actually 2 tools that we use. There's an ATOS score and another readability text tool, and we plug them in there, and it gives us a general grade level. And then also one of them gives lists of multiple syllable words or average sentence length, and tells us what we can change to make it a little bit more understandable.
All right, thank you so much.
Of course.
Our next presenter is Chen. He worked with our associate company, Gene Capture. This was a new internship this year. We've never had an industrial design intern. Chen is from Auburn, and we were happy to have a new experience this summer. So, Chen?
Hi, I'm Chen, and I come from Taiwan. And I briefly introduce myself. And I currently pursue my second industrial design master degrees at Auburn University. And I have 3 years of working experiences focused on industrial design. And I'm also a freelance motion graphic designer and help customers making a commercial video, including a 2D, 3D animation, and filming. Next. And Grant Thompson, she is my mentor in this summer, and he's also a COO in the Gene Captures. Yeah, next. Next. Okay, and my current project is sponsored by the United States Air Force, and the goal is for Gene Capture is to design a new rapid infection detection platform. And my main duties are doing design research and design the device and its interface. Next. And at first, I visit the company to develop a relationship with the team members. And next, I explain the industrial design because I'm only the designer in this team, so they don't have the experiences of collaborating with designers. So I have to do that. Next. And at first, I present the initial user journey map and the flow charts, and there are the 2 different design approaches to analyzing the user behavior, including their action and emotion, try to find their pain points, and that's our opportunities. Next. And the team members introduced their solution of infection detection platforms and prototype. You can see the pictures that they are including the prototype of the device and the cartridges. Next. And then because we will plan to conducting the user interview, so I prepare the user interview scripts, and then I create more detailed flow charts. I revised the new version by getting some feedback from my team members. Next. And I analyzed some competitive product in the current market and try to find some references and to analyze their disadvantages and advantages. Next. And I propose some solution by sketching, such as how to deal with the waste and designing a package for the supplies and designing a sample loading station on our device. Yes. Next. So the design research goal is to make sure the product requirement meets the user needs, and we will use the research data to create a persona and the user journey map and the user flow chart. Yes. So and finally, I really thank the all team member in the Gene Capture and Michelle. Yes. Thank you.
If anyone has any questions, we would welcome those for Chen. And we are going to go ahead to our next presenter. Next we have Christina Stacy, who worked with our communications team. Big team effort working with Christina this summer. So welcome, Christina.
Hi, everyone. Thank you, Michelle. I'm so honored to not only have been chosen for this internship, but to have done it at HudsonAlpha. I think it's a testament to the impact that HudsonAlpha is making on not only students, but the world around them, as it's my last week and it's so hard to leave. I graduate in 2 days from the University of Alabama, and I think I have learned more over the course of 3 projects in 1 summer than I have in 3 years of school. So again, a testament to that impact that HudsonAlpha is making. Next slide, please. So I worked on 3 projects over the course of the summer, as I mentioned before, and those were focused around graphic design. The employee interviews kind of helped me with improvisational thinking skills, and then website analysis was a good area of growth for critical thinking. Next. So the first thing that I'm going to talk about is the graphic design portion. This was probably the longest and most strenuous project, but it was also the most team-oriented. I was able to work with some really, really talented team members of the comms team. Some areas that I'd like to highlight in this, as I mentioned, teamwork, but just the value of growth in something. I think that if something is ever easy, then there's probably a lack of growth associated with that. And so I spent many, many days frustrated with my lack of knowledge in Adobe design platforms, but I'm not a graphic design student. And so frustration is normal, and that frustration can lead to growth. And so there's positivity that comes out of that. And I think that's the important thing that I learned, and that the mindset of being a lifelong learner is what's important when you have that frustration. And then, of course, the graphic design portion. But if you look at the graphic on the bottom right, the 4th of July graphic, you may have seen these on Instagram. I made this, and this was the one that I got frustrated with learning how to make fireworks, watching various YouTube videos, getting tutorials from team members. But this was the most beneficial part of teamwork that I learned, because the last day, it was, I think, July 2nd or July 3rd, when I gave it to our social media guru to post, it looked horrible in my opinion, and she was kind of giving me some critique. I sent it to our graphic designers, and they had it up in no time and did it without question. So I think, again, the value of a team is amazing. And if you have no graphic design experience, just know that those people are heroes. They're amazing. Next slide. This project was super interesting. It kind of gave me a little bit of insight into HR. So in an attempt to help prospective employees understand what HudsonAlpha stands for and the culture behind HudsonAlpha, I had the opportunity to interview, reach out, and then interview some of the new hires. This is just one that I focused on. The link there at the bottom, if you'd like to check out those employee interviews, they're on the website posted. But what I gained from this experience is, as a almost recent college graduate, I don't think I'm alone in saying that my graduating class is experiencing this new transition into a new normal. And we don't quite know what our work environment is going to look like. And so the fact that I had the ability to learn how to communicate using only 2 platforms over the summer, Zoom and email, I think is pretty amazing. And I was able to develop a relationship with not only the team members, but these new hires, and just get some insight into their career paths and their work experiences. And then I was also in that able to learn how to manage time efficiently and manage a project efficiently. Next slide. And this last one, the comms team is working on kind of vamping up the HudsonAlpha website and just going through it and figuring out what we can do better with it. So I had the opportunity to perform a critical analysis of the website. And this kind of gave me this understanding that not all members of an organization or a situation have the same accessibility to a certain topic or subject. And along with that accessibility, not all of them have the same background knowledge on it. And so I was able to take the perspective of a physician approaching the website, a researcher approaching the website, a parent, a student, an educator. So getting that unique perspective of all of those types of backgrounds in one gave me this transferable skill that I can take into my career path of taking a step back and understanding how everyone perceives the situation. Next slide. So thank you, not only to Michelle and to HudsonAlpha for allowing us to participate in this new remote internship, but to the comms team and Laura for all of your wonderful support and help through this internship. And I guess I'm open for questions if you have any.
Great, thank you, Christina. Appreciate it. All right, so if you still have questions for Christina, don't hesitate to type them in while we go on to our next presenter. Jay Herron from Auburn University worked with our clinical services lab also. He was mentored by Drs. Elaine Lyon and Melissa Kelly.
Hey everyone, my name is Jay Herron, and I'll be talking about my research this summer determining the metrics for clinical utility of genome sequencing. Clinical utility refers to the ability of a test result to provide information to a patient. So in the world of clinical testing, clinical utility must be demonstrated for insurance companies to pay for testing. To this day, there is limited data regarding the clinical utility of genome sequencing. So for my project, I wanted to identify metrics of clinical utility by gathering preliminary data on the clinical utility of genome sequencing. There's little evidence actually showing the utility of sequencing, but anecdotal evidence shows that genome sequencing can be useful. While these success stories are fulfilling, they are of limited practical use as evidence of clinical utility. Insurance companies must be presented with quantifiable data. Next. First, I determined possible metrics of clinical utility. Then I data mined patient records, sequencing reports, PubMed, and other literature resources to gather the data to present these metrics as means of clinical utility. Because the data was gathered retrospectively, information was scattered across these sources, making this process manual. When finished data mining, I wanted to transform the qualitative nature of this data into a measurable quantitative data. Next. The first metric to look at is diagnostic yield. This number shows the percentage of cases diagnosed. The overall yield was calculated and the diagnostic yield per disease category to see if one phenotype had a higher likelihood of getting a diagnosis. The next metric is treatment availability. Clinical utility can be demonstrated by revealing a treatment plan to a patient. Some diagnoses, such as autism, listed resources as part of the management, such as physical therapy. I did not categorize these resources as a treatment of the disease, but more of a means to improve quality of life. Additional or confirmatory testing was considered as another metric. For example, a diagnosis of familial breast cancer predisposition can change the age to begin mammograms or the frequency. And lastly, the impact of the diagnosis on family planning is measured by how many diagnostic variants are inherited or de novo. Next. This pie chart on the left has cases split into 5 categories: diagnostic, possible, partial, possible, partial, and not diagnostic. The diagnostic yield of 22% does not account for the possibles and partials. The yield could actually be around 30% if some of the possibles and partials were really diagnostic. Regardless, roughly 1 in every 3 or 4 patients are receiving a diagnosis, ending their diagnostic odyssey and proving clinical utility. So this bar graph on the right shows both the diagnostic yield and just the raw number of confirmed diagnosed cases in the blue, along with the undiagnosed cases in the orange. It's broken down by disease category associated with the patient's phenotype. Those percentages come from the diagnosed cases in the blue divided by the total cases, which is the total height of the bar. So from this graph, you can draw that patient phenotypes associated with neurology, developmental delay, or abnormal tone have a higher likelihood of getting a diagnosis. Next. This pie chart on the left shows that a third of diagnostic cases have an available known treatment specific to the disease. On the right, you can see that 76% of diagnostic cases have available additional testing. These are the two lighter shades of blue. From a clinical utility standpoint, 36% of the patients whose diagnosis has an available additional test haven't actually performed that test. So upon diagnosis, these patients can now do these tests to better their care. Next. Families can make informed reproductive decisions in knowing that the disease-causing variant is inherited or de novo, which is a new mutation. Of the diagnostic cases, 39.5% found an inherited variant, 47.4% found a de novo, and the remaining cases are unknown because the parents weren't available for testing. Next. So from these stats, we can conclude that genomic sequencing has clinical utility in at least 3 ways. 1, by genome sequencing, 30% of patients finish their diagnostic odyssey. 2, families can make informed reproductive decisions knowing if the disease-causing variant is de novo or inherited. And 3, 1/3 of diagnoses have an available treatment plan catered to the disease. Next. Some limitations of this research. So sequencing singletons or the genome of just the patient at focus can be disadvantageous because without the family's genomes to compare to, it becomes more difficult to find that diagnostic variant and impossible to know if the variant is de novo or inherited. And as I said before, the data mining was done retrospectively and manually. Perhaps some sort of search engine could be developed that scans through a patient's records to find the answers to these metrics much easier. Also, the CSL doesn't have direct access to the patient or medical records. For future directions, I can see the clinical utility of genome sequencing being demonstrated by these metrics with respect to before and after receiving a diagnosis. Time in healthcare, money spent on patients' healthcare, number of meds prescribed, and frequency of episodes due to the condition. Next. I'd like to acknowledge the following individuals for a great summer. I'm really so thankful for this opportunity and the time that all these people have invested in me. Next. Any questions?
All right, thank you so much, Jay. Don't have any questions typed in right now, so we will move on ahead. But if you think of a question for Jay, don't hesitate to type it in. Right now, I'd like to welcome Jordan Manchego. Jordan's from University of Alabama in Huntsville, and she worked with our plant faculty members, Dr. Alex Harkis and Dr. Sarah Carey.
Hi, my name is Jordan Manchego. I'm going to the University of Alabama in Huntsville, studying biochemistry. I'm going to start my last year there. And this summer, I've been working in Dr. Alex Harkis's lab, developing an automated duckweed phenotyping pipeline using plant CV. Next slide, please. So first, why study duckweed? Well, actually, it's a really amazing plant. So there's 37 species of this angiosperm. And while it can produce flowers, it clonally propagates at exponential rates in very little time, super rapidly. So this time-lapse video below shows just how quickly Spiraldella duckweed grows in just 3 days. And this time-lapse video was made in collaboration with several scientists at the Danforth Plant Science Center, including Blake Myers, Kirk Zimick, and Kevin Cox. And duckweed accumulates biomass at really high rates and can serve as an indicator of environmental health. The Food and Agriculture Organization of the United Nations has found that duckweed contains up to 43% protein content, along with many essential amino acids. This makes it a great candidate for an affordable and sustainable dietary supplement for the health conscious. Next slide, please. Oh, go back one. Yeah. My work this summer has been focused on using the Danforth Center's Plant CV image analysis tools to process individual frames from this video of Spiraldella duckweed growth over 72 hours. Workflows provided by Plant CV are sometimes tailored to plants that have stalks and stems, but duckweed provides a challenge because it's a flat plant that grows on the surface of water. So I use OpenCV to extract individual frames from the time-lapse video, then Jupyter notebooks to streamline the Python scripts at each line so they can be easily recreated and manipulated to work better on flat plants like duckweed. So far, I've improved the process for analyzing the surface area of Spirodella duckweed by altering PlantCV's visual image system workflow to create an overlaying mask that by thresholding the color hues of the original image instead of the color saturations. I've also refined the organization of output images, so the user has a lot more flexibility when they're naming like numerous image frames and output files all at the same time. I did this by using bash scripts, so it's eliminated the need for including the timestamp and the model of the camera in the input image's name. So by simplifying the process of using PlantCV, workflows, and creating easy to follow directories, I'm trying to make this program easier to use for students and scientists who might not have a background in bioinformatics. Next slide, please. So just in the last few days, I've been able to isolate the surface area data from the entire time-lapse image set to produce this animated graph that you see on the right. So this incredible data set precisely and automatically quantifies the surface area of this duckweed at each hour of its growth. As you can see on the left, you've got the original time-lapse, and on the right, you can see its exact surface area increasing. Next slide, please. And in the future, I will use my scripts to create automated pipelines that will analyze many plants at the same time, making simultaneous experiments way easier. My intention is to organize an automated cascade of Python and Bash scripts that will process images of duckweed in real time as they're captured. There are even tools available that could alert scientists to significant phenotype changes as they occur via email, Slack, or text messages. My next steps will be first converting my surface area data from pixels to millimeters squared, then defining high contrast regions as leaf overlap, as you can see in the image on your right. Overlapping regions may then be considered additional surface area for more accurate biomass calculations. I will also improve the PlantCV morphology workflow to work better on flat plants that don't have the visible stem. And I hope to make my workflows public to encourage collaboration and improvements so they become easier to use under various conditions and operating systems. Next slide, please. So thank you for your attention today. And thank you to these individuals who made my research possible this summer, including the entire Educational Outreach Department at HudsonAlpha. This summer has been actually really smooth for the online transition, and I really appreciate everything they've done for us. I wish I could see you all in person, but if anyone has any questions, I will do my best to answer them.
All right, thanks, Jordan. We have a question from James. Jordan, how are the phenotypes identified? Is this a learned feature, i.e. machine/deep learning, or something defined by a researcher?
So the researcher would have to use machine learning to threshold certain values. So let's say maybe the duckweed's got yellow splotches on it, and because of maybe like dehydration. That would have to be identified and defined in the program. And then you could threshold those certain yellow hues to be quantified in a certain way. Like say a certain amount of surface area covering the duckweed would be, that value would be like isolated. But I guess it would be more machine learning.
Okay. All right. Thank you so much, Jordan.
We have a second question in the chat from Megan. Where does duckweed grow?
So duckweed is found on mostly standing water, lakes, ponds, and it will usually carpet the surface if it can of these bodies of water. And because it grows so quickly, like it can just really carpet it. And it's, I believe it's found in so like all around the world, mainly warm climates, but if it gets cold and like the pond freezes, duckweed's able to sink to the bottom, freeze, and then come back up when the water warms up again. But I believe it is worldwide. It's found in many, many places across the world, including here in Alabama.
All right, Jordan, I have one more question. How does duckweed taste?
Oh, so I've never tasted it. I wish I had. I actually cannot find it here in Huntsville, so I have not tasted it myself. But Dr. Harkis says it tastes very chlorophyll-y, and I'm not sure how to describe that to you. My guess would be, I guess, maybe close to spinach, because it'd be really rich, and I have no idea, honestly. I wish I could tell you.
We'll have to all explore that as our next culinary adventure. Thank you so
Exactly.
much.
Incorporate it into some pasta. Thank you so much.
So our next presenter today is Case McCormick from the University of Alabama. Case worked with the researchers in Dr. Greg Cooper's lab, and I'm happy to have him share with you his research today.
Hi, everyone. So for my summer project, I created a software pipeline that identifies disease-causing mutation in mitochondrial DNA. Next slide, please. So my lab has mainly been focused on nuclear genome mutations, and they recently began pioneering the mitochondrial genome. As such, they are in need of more tools to help streamline the mitochondrial genome analysis. And my goal was to create a tool that makes that process easy and efficient for them. All right, so the mitochondrion is the energy production system of the cell, and it's necessary for cell metabolism. It contains its own genome that codes for necessary proteins involved in energy production. Therefore, a genetic disorder of the mitochondrion will inhibit cell metabolism and can cause inflammatory conditions in the patient. Probably the most important point here is that the mitochondrion contains many copies of its genome. So we're not only asking, which mutations are likely to cause disease, but at what frequency do they occur in all the genomes in a person's mitochondria. And then lastly, we inherit our mitochondria from our mother, so they're present in the mother's egg cell. So I'll discuss the significance of this later. Next, please. So to detect mutations, we have to collect the blood samples in lab, ideally from a patient and their mother. And then once the DNA is extracted from that blood sample, we can convert it to computer data with sequencing technology. Next. So here is kind of the whole computerized process of DNA analysis, or variant analysis at least. I won't go into too much detail here, but basically each circle is a different file type and each arrow is a different process we apply to that file type. So the end result file is a variant file which lists all mutations in a person's blood sample. And then I'll hit the next button, please. And then my program kind of handles these last few processes and stores our variant data in the database. Next. So here is the variant file that I look at. Each line is associated with a different mutation. And then there's a bunch of other relevant data, like, we have other resources that can report whether this variant is pathogenic or confirmed to not be pathogenic. And so that's all listed here. And so I will extract all of these data fields and store them in the database, but not each one is relevant to the scope of this project. So next, please. Let's take a look at one of these. I'll hit the next button. So this first data field is the position. So where does the mutation occur in the genome? Next. The variant is the reported mutation. So at position 263, there's a guanine. When in the standard healthy genome, there should be an adenine. Next, please. So the depth is basically out of all the genomes we collected in our blood sample, this particular mutation occurred on 124 of those. And next, the most important is the frequency. So out of those 124 genomes where this variant or mutation occurred, this is the frequency of the depth, or the ratio of the depth to the entire person's mitochondrial collection of mitochondrial genome. So a frequency of 1 says that this particular mutation is present in every mitochondrial genome in this person's body. Next. So my program methodology, basically I used Python to program everything. It's popular and it supports all the functionality we need. But probably the most important tool here is the database. So by using the database, we can more easily and efficiently analyze our variant data. And I chose SQLite because it's simple and easier to use than most other big databasing systems. Next. So once all of our variant data has been collected from our blood samples and stored in the database, we can use queries to select out of all the variants we have, which ones are likely to be pathogenic. And this one is where the mother's genome comes in. So let's say a child exhibits symptoms of a mitochondrial disease, but the mother doesn't exhibit symptoms. If we can collect both genomes and compare them, we can narrow down our search from, on average, 100 variants to probably 5 or less. So that is where the efficiency comes in for the researcher or the doctor. Next, please. So the benefits of my program, it automates a lot of our variant analysis processes, and also automates the external data cross-referencing. So there's a couple other labs that research mitochondrial genomes, and my program will kind of search through their data set and compare results. So not only that, but my program is lightweight, modular, and it's pretty fast. Makes the comparative data analytics process super easy for my lab. All right, next. I want to say thanks to Dr. Cooper and Dr. Hyatt for guiding me on this project, James and David for the technical help, and Stephanie for just being cool.
All right, thank you so much, Case. Again, I apologize for crashing in the middle of your presentation. It's hard enough to do this anyway without having that disruption, but you hung in there and you did an excellent job. We don't have any questions from our viewers right now for Case, so we will move on to our next presenter. And this is our last presenter today. I'd like to welcome Susannah Putner from University of South Alabama, who also worked with our genetic counseling team, particularly with Kelly East and Veronica Grief.
Okay, so first of all, I also have a storm coming, so please just let me know if you can't hear me. But I will go ahead and get started. Okay, so like Ms. Michelle said, my name is Susannah Putner, and I had the privilege of being one of this summer's BioTrain genetic counseling interns. And my research project for this summer involved performing a qualitative analysis on a population-based screening program called Information is Power. You can move on. Okay, so first of all, what is Information is Power? As I said, this is a population-based screening program that was launched in 2015 as a collaboration between HudsonAlpha and Kairos Genetics. It offers free and reduced cost hereditary cancer screening to residents of 5 North Alabama counties. And in particular, it's directed at low-risk populations, so those individuals that would likely not be referred for genetic testing based on personal or family history. And to date, over 5,000 people have participated in the initiative and therefore learned more about their own genetic risk factors for cancer. Next. Okay, so our research questions were concerned with finding out what were the characteristics of the participants who elected this testing, how do they interpret their results, what impact did they perceive the results to have on their life, and finally, what were some of the benefits and limitations that they considered to be part of a population-based screening model such as Information is Power. For our methods, we interviewed 19 consenting participants, and then 3 independent coders, including myself, reviewed and reconciled each of these transcripts in order to perform a qualitative analysis and induce some common overarching themes. Next. Okay, so the first thing we were looking at is what were the characteristics of these self-initiated participants. We found that 63% stated they believed in patient autonomy, or that patients should be able to make their own healthcare decisions without necessarily having a physician standing as sort of a gatekeeper. We found that 84% were communicators or discussed.
Well, it seems like we might have lost Susannah. Perhaps her video has frozen. While we're pausing just a minute, I do have a question for Case. Case, if you are able to answer a question. We had one from James.
Yes, so Elaine asked one first, and I wanted to say it out loud for everyone.
Okay, go right ahead.
Elaine asked if I was able to apply this to cases. So I had a 54 genome sample size, which I tested it on. So each genome contained on average 100 mutations, and my program was able to narrow this down to about 5 or less. So depending on the type of data or variant caller we used, this produced more or less mutations. But my program was able to kind of narrow this down and pick out, a subset of which are likely to be pathogenic or highest result pathogenic.
Okay, and Thanks, Case. The next question from James was, have you found any samples with mixed or multiple mitochondrial sequences?
I'm not quite sure what you mean by mixed or multiple, but from the blood sample, you know, once the mitochondrial DNA is extracted, we have many genomes, and each genome is going to be slightly different. They're not all going to be the same. But the program compares all variant possibilities, all mutation possibilities, and has a lot of associated data with that. So out of that associated data, we can kind of pick out which ones are most likely pathogenic.
All right, great, thank you. Susannah, welcome back. I'm glad that you were able to come back in, and we will invite you to continue.
Okay, sorry. Were you all able to hear through this slide?
We heard the first point you were making on this slide.
Okay, I'm very sorry. Hopefully I can stay in this time, but I will move on. Okay, so yes, 63% thought it was very important that patients should be able to take charge of their own health. We found that 84% communicated their decision to take part in this initiative with friends, family, or a healthcare provider. And then 84% did specifically state they were information seekers and just wanted to know more about their own health. Next. Okay, so as far as beliefs around cancer, the results were mixed here, but we did have about half the participants state that they believed there was a large genetic influence. About half stated they believed there was a large environmental influence, but notably, the majority of participants did believe that there were preventative measures that they could take to help mitigate their own risks for developing cancer in the future. Next. Okay, as far as impact of results, we found that the participants in general had the results provide relief, but really no major impact. But an important thing to note is that many of them did state that regardless of what the results said, they did intend to continue with appropriate screening, which of course is extremely important. Next. Okay, as far as the perceived benefits, an overwhelming majority found that patient empowerment was a very good benefit for programs such as Information is Power. Again, this is letting the patients take charge of their own health. Another big benefit that they perceived was convenience, so the process being relatively easy, fast, and of course, cost-effective. The majority also stated that they believed they had an increase of knowledge of cancer or the genetic testing process in general, and just under a quarter found it beneficial that they did not necessarily have to share the results with a third party. Next. Okay, and finally, the perceived limitations. So it was noted that about 70% of those interviewed voiced concerns about potential misinterpretation of results and stated that it could be beneficial to have a physician present to disclose these results and make sure that the patient clearly understands what it means for their risks. Just under half also stated potential concerns for psychosocial limitations, so perhaps increased distress or anxiety. And then about a quarter said they had some concerns about data safety and whether their results might be shared with a third party, such as an insurance company. Next. Okay, so what are the next steps? The information will be written up and constructed into an academic article and published into a relevant academic or scientific journal. A possible addition to the research would be conducting interviews with physicians to gain insight into their perspective on Information is Power or similar programs. But ultimately, we hope to use this information to not only improve the current methods of Information is Power, but also to help guide other organizations that are looking to launch their own population-based screening model. Next. And finally, of course, I would like to say thank you to these individuals that made this research possible and that allowed me to have such a great and impactful summer here, in quotes, at HudsonAlpha. And with that, I'll be happy to take any questions.
Thank you very much, Susannah, and thank you for coming back. We're happy to hear the end of your presentation.
Sorry about that.
So I don't have any questions right now for Susannah. Of course, if you have some, please do type them into our question and answer box. I do have a clarification for the question that James asked for Case. So just to remind you, James had asked about the mitochondrial. I'm going back to his questions now. What did he ask? Did you have any samples with mixed/multiple mitochondrial sequences? And he clarified that to say, an individual is expected to have one version inherited from the mother of the mitochondria. Did you have any with multiple versions, Case?
Yeah, so the query that I showed only picks out the ones that were present in the mother to a difference of 10% between the frequencies, but it also picks out the ones that were not present in the mother. So it can do kind of all the above. And so the databasing technique actually makes picking those separate sequences out very easy. And that's why the database for this project is important.
All right. Thank you, Case. I also have another question from Rick. Case, was the frequency always 1.0 for the variants you found?
Not at all. So that particular variant that I chose was just a random one from my output. But we did have some, so the ones we look for to be likely pathogenic are greater than 0.95. So that particular one was actually not pathogenic at all. It was present in the mother as well. So that's likely to, rule that one out.
All right, thank you, Case. My next question is for Susannah. Susannah, James would like to know, I might have missed this, but what was the breakdown of number of people interviewed that had negative results versus an informative result?
Okay, so that's a good question. Out of the 19 individuals that we interviewed, I don't want to try and give an exact answer here, but I do believe that only maybe 2 to 4 of them had positive informative results. But I mean, I don't want to say informative because even if technically they got a negative result back for risk factors, that is of course still informative. But as far as they did have a genetic predisposition present, only a couple of those interviewed.
All right, great. Thank you, Susannah. I don't have any more questions at this time, so we'll go ahead and wrap up this session. I so appreciate those of you that were able to hang in there with us through our disruptions. Storms come at the worst times sometimes, but we're very proud of all these interns and just thrilled at the way that this summer went. Of course, working remote was an experiment, and I would say it succeeded, and I hope that we'll be able to continue this as needed in the summers to allow more students to have some more options. So we appreciate all of you viewing with us, and hope that if you're interested in the BioTrain program, that you'll go to our website to get more information. Thank you and have a great day.